So dear students, in previous video, you come to know about the difference between genetics and genomics. So now in this video, you will learn about the genomics and microbial genomics. So this is the part 2 of the first lecture. So earlier you come to know about the cell biology, and about the location of nucleus in the cell and location of genetic material inside the nucleus. Then this genetic material is in the form of DNA or RNA in case of prokaryotes. So you have seen that there are the DNA sequences on the chromosomes. So when you study the person's gene or the genome, including with the interaction of those genes with each other and with person's environment, that is the genomics. You learned about the genomics, that is the about the genome of the person as well as about the environment, about its environment. So that is known as the genomics. So in genomics also, if you study about the bacteria, viruses, archaea and fungi, which are, you can say, the microbes, then it is named as the microbial genomics. So microbial genomics, it's a interdisciplinary field of science that is focusing on the structure, function, evolution, mapping and editing of genomes of the microbes. So it means study of entire genetic material of microorganisms including bacteria, viruses, archaea, and fungi, etc. So, by analyzing these sequences, researchers can understand how microbes evolve, they cause the disease, they interact with their environment, and can be harnessed for the biotechnology. So, microbial genomics means these genomes are widely variable and reflect the enormous diversity of bacteria, archaea, and the lower eukaryotes. So plants-based microbes, they play very critical role in the agriculture and food safety and security and in maintenance of ecosystem balance. Some of these diverse microbes, which include viruses, bacteria, omicides, and fungi, they cause the plant diseases, while others prevent disease or enhance the plant growth. So less than 6% of microbes whose genome sequence has been completed are plant-associated microbes. You can study the genome of the bacteria, or you can study the genome of the viruses or the fungi, etc. So what is the importance of microbial genomics? Actually, microbial genomics has provided a foundation for the broad range of applications, from understanding its basic biological processes, then the interaction of host and pathogen, and protein-protein interaction, and also to discover the DNA variation that can be used in genotyping or forensic analysis, and the design of novel antimicrobial compounds. So these are very important for the designing of antimicrobial compounds. There are other genomics also, in addition to the microbial genomics, like structural genomics. So when you are mapping and sequencing the physical nature of DNA within the genome, this is known as the structural genomics. Then functional genomics, when we investigate the roles of genes and how their expression influences cells phenotype, means how these are expressed, what role or what function they are going to play, then it is the functional genomics. Then comparative genomics, it means we are comparing the genomes across different species or strains to identify evolutionary pattern and unique genetic signatures. So when we compare the genome of different species, then it is a comparative genomics. Then in the next more evolved form, that is metagenomics, which I will uh, give the details in the next video. So studying the collective genome from the entire microbial communities directly from the environmental samples and bypassing the need for the laboratory cultivation. This is a metagenomics. So we are focusing on the genome from the entire microbial communities from the environment sample. That is the metagenomics. So that will very helpful in the prevention of disease and to study the spread of disease, how the disease spread by these microbes. So it is very important to study the metagenomics to understand the nature and